the very beginning, kind of empty, and then as the years have gone by, the lab has changed a lot. Praneeth's been there through all of it. And so I was thinking back, and I almost forgot, I almost blocked out these memories that I had of Praneeth when I first met him, because it's been such a long time and he's grown and developed so much. But when I first met him, he was clearly very intelligent and he had a really strong engineering and computational background. But I'm going to be completely honest here, I was a little worried about how he was going to do in the lab. He's a little bit stubborn, confident, unorganized, rougher around the edges, and I wasn't sure if he was going to be a hard worker. And so, he did this first rotation in the lab, and I actually didn't think it was going to work out. And I remember, having this conversation, and I was just so surprised that he wanted to join the lab because he hadn't seemed very serious about the lab and that he hadn't really shown up a whole lot. And so I told him these concerns, concern, are you going to really come and do work, and what's going to happen? I was not really sure how to handle it. And the lab was just so young. There's nobody in the lab. It's just an empty room. So I was like, okay, well, I guess you can do a second rotation. And I am so glad that I gave Praneeth the second chance, because he completely proved me wrong. I was wrong about pretty much everything. Praneeth isn't smart. He is a genius. And Praneeth is one of the most hardworking and motivated people that I've ever met. He's just absolutely dedicated. And I find myself often saying, Praneeth, you should really go home and take a nap. He'll just work. There's just no end. And I feel like I almost have to hold him back sometimes. And if I left him to his own devices, he would almost certainly cycle to a nocturnal circadian rhythm. But these days, he's possibly the most organized person, and we've all really come to depend on him. And I was thinking about doing this introduction, and I thought, how could I do this without mentioning Anna? And at first, I think Anna will, if you dig through your memory, you'll remember also that I almost felt like it was a favor. Oh, Anna, will you help me train Praneeth? Da, da, da. But then, really soon, Anna and Praneeth formed this beautiful relationship. We all spent, lots of late nights working together. And for the past couple of years, I feel like I've been learning from Praneeth. Praneeth has been teaching me, and I'm just asking him for advice and taking notes. And I'm sure soon-to-be Professor Anna Baylor, when she starts up her own lab, I think she'll agree with me that neither of us would be where we are today if not for Praneeth. And I can't believe how much I've learned from you. And I always hoped Praneeth would turn out well, but he really surpassed my wildest dreams in terms of what I even imagined. This is just a picture of Anna and Praneeth ringing the data bell. This is the bell that we have in our lab to ring whenever something good happens, to crystallize this moment. And then, of course, their co-first authorship, Praneeth's paper with Anna that he landed the cover on of Nature. And Praneeth always loved challenges, and I always presented a number of challenges to him. He was always up for it. Whatever it was, didn't matter how hard it was. If we do this, if we pursue this line of questioning, it's going to be a lot of work. He always wanted to do it. He always wanted to do, the path of the greater challenge. And he did a great job leading this team, especially during manuscript revisions, which was a very stressful time. And I think during this stressful time, Praneeth really put forth a heroic effort and became uber organized, an amazing team leader, a great communicator, and blossomed into what I believe, I see him as a true visionary. And so he's been extremely productive, has been also a very generous, supporting team member, and has worked closely with another postdoc, Gwen, and also what he calls Team Retrobeads. And only today, when I was putting this picture together, I was like, wow, it's like Praneeth and all these ladies. So anyway, he's also, in addition to being a really hard worker and really admired, he's also got, this personal touch. So from, I think he's shaving, holding the baby, hanging out in the lab. He's been great. And what some of you might not know is that Praneeth is also a competitive ballroom dancer. This is an important development that he underwent during the lab. So he's right here. This is him this past weekend. Is it last weekend? So maybe 2 weeks ago. So anyways, this is Praneeth. How far is he going? Sure. I'll give it 10 more seconds. Everybody enjoying this? Okay, so anyways, I could not be more proud of Praneeth. I really look up to him now. And there's nothing else I could possibly ask for. And in fact, I know he's destined for a spectacular career in research, and I just feel lucky to have been part of the journey. So with that, I'll leave it to Praneeth to tell you about his thesis work.
Thank you for that amazing introduction, Kate. Thank you everyone for coming. Today I'd like to talk to you about basolateral amygdala circuits for differentiating positive and negative associations. So I grew up in India and I went to school there. So we had a person ring a bell to indicate passage of time, so to speak. Some periods were more interesting than others. So one of them was called the long bell, and when he rang it, it meant lunchtime, so I was really happy. So I really liked the long bell. If the bell rang twice, in the morning we were supposed to assemble in a courtyard. As Kay mentioned, I'm not a morning person. And if I hear that bell on my way to school, it means that I was going to be punished in ways not entirely legal in the US. I didn't really like that sound. Anything that's attractive is said to have a positive valence, and anything that's aversive is said to have a negative valence. Today what I'm going to try to talk to you about is a little bit about how the brain differentiates between positive and negative associations. How does the brain achieve this? So perhaps there are different regions in the brain. Some of them encode positive valence, some of them signal negative valence. However, if you take a closer look, if this beautiful rounded rectangle represents one brain region, then in many cases you will find neurons that selectively signal positive valence and neurons that selectively signal negative valence. These are just a few examples of brain regions that contain intermingled populations of neurons which signal positive and negative valence. And if you actually go look at the numbers and contrast between the two, there is a mixture in every single brain region of neurons that represent positive and negative valence. So perhaps we should be looking at populations of neurons within a brain region rather than looking at different brain regions for encoding positive and negative valence. So today I'm going to talk to you about basolateral amygdala. Why basolateral amygdala? Because Keta is my advisor and she loves the BLA. No, but seriously. BLA is a good region to study valence coding mechanisms because of these 4 reasons. BLA has neural responses encoding valence. It receives convergent stimulus information required for making these positive and negative associations. And learning changes plasticity onto inputs in the BLA. And BLA sends axons to has divergent pathways for expression of behavior. So let's go through the last one in a little bit more detail because it's important for this talk. So if we look at basolateral amygdala projections to the centromedial nucleus of the amygdala, first, if we stimulate centromedial nucleus of the amygdala, we evoke aversive behaviors. Then if we disconnect BLA from CM, how do we do that? In a mouse, since these regions don't send axons to opposite hemispheres, you lesion BLA on one hemisphere and CM on the other hemisphere. So technically the mouse has both regions, but they're not connected. So if you disconnect these 2 brain regions, then fear expression gets abolished. So then let's talk about another projection, BLA projections to the nucleus accumbens. Stimulating BLA projections to the nucleus accumbens promotes self-stimulation, which is a positive behavior. And inactivating BLA in AC pathway reduces behavioral response to sucrose, which, so it's a bidirectional modulation of reward seeking. So perhaps projection target for BLA neuron plays a key role in valence processing. So what do I mean by that? I mean, so I said that BLA has two different functional populations, one selectively responsive to positive cues and one responsive to negative cues. And I also told you that BLA projections to NAC and CEM might be involved in opposing behaviors. So now we're going to investigate the effect, the role of projection target defined BLA neurons in valence processing. So before we do that, we need to know if these two cell types are distinct. And what do I mean by that? So we could have a scenario where these two cell types are distinct, or the same neuron can send axons to both of these brain regions. So in order to investigate this, we used a viral approach where we expressed two fluorescent reporters in either NAC projecting BLA neurons or CEA projecting BLA neurons. One of the reporters was M-cherry, and it was fused to a protein called synaptophysin. Synaptophysin gets trafficked to the synapses, and therefore only the synaptic terminals are going to light up. Here's a representative image on top of EYFP fluorescence from this protein, and on the bottom is fluorescence from synaptophysin M-cherry. You'll see little, it'll look like little dots. So if we look at collateralization among NAC and CEM projectors, if we express synaptophysin M-cherry in NAC projecting BLA neurons and look at NAC and image in NAC, we see more terminals, a lot more terminals than in centromedial nucleus of the amygdala. Conversely, if we express synaptophysin M-cherry in CEM projecting BLA neurons and image in CEM, then we'll find a lot more synaptic terminals than in the NAC. So, what am I trying to say? I'm trying to say that I'm trying to distinguish between these two models, and as always in biology, the truth is somewhere in between. But we can kind of estimate the proportions of the gray neurons relative to the red and green cells. And if we inject retrograde tracers into NAC and CEM and do this estimate, it turns out that the gray population is only about 5% of the neurons of this entire population. So moving forward, I'm going to use this notation because they're largely distinct. So now I showed, so there are two distinct populations of neurons in the BLA. Do they do two different things in terms of evoking positive and negative behaviors? So perhaps stimulation of NAC projectors evokes positive behaviors and stimulation of CEM projectors evokes negative behaviors. In order to do this, we used a viral approach. Viruses are essentially like FedEx packages for delivering genes and expressing them in specific populations of neurons. And different viruses infect different parts of a neuron, for example. We use a rabies virus, which when gets taken up at the axon terminus, and whatever the payload gets expressed in all the neurons that project to wherever I injected the rabies virus. So in this case, here's an example image of in an animal where I injected RVCHR2 venous in the nucleus accumbens. And sure enough, I found expression of neurons in the basolateral amygdala. One of the proteins that we are expressing in these neurons, in addition to the fluorescent reporter EYFP, is channelrhodopsin. Channelrhodopsin makes the cells sensitive to light, and upon shining light on these neurons, we are able to activate the neurons. So I made brain sections from these cells from a mouse expressing RVCHR2, and I got hold of one of the neurons. When I gave it blue pulses of light, it showed robust firing after 5 days of viral expression. So now we can take advantage of this and ask the question whether if we stimulate NAC and CEM projectors in the animals, do they like it or do they hate it? So to see if they like it, we use an assay known as intracranial self-stimulation. In this assay, the mouse can choose between two different nose pokes. If the mouse makes a nose poke in one of the ports, he is going to receive a laser stimulation, which is going to activate either in one population of mice, either NAC projecting BLN neurons, in another one CEA projecting BLN neurons, and another one would be a control. So in a mouse where we stimulate NAC projecting BLN neurons, x-axis here represents time and y-axis represents how many nose pokes he makes. We can see that the mouse makes a lot more nose pokes in the active port where he receives laser stimulation as opposed to the inactive port. And if we look at the population data, we see that the mice which receive stimulation to NAC projectors like it. So therefore, stimulation of NAC projectors is sufficient to cause self-stimulation. Conversely, we used a real-time place aversion assay to see if stimulation of CEA projectors is aversive. Here, in this assay, we put a mouse in a rectangular box, and when the mouse goes to one side of the box, shaded in blue here, he receives laser stimulation of a CEA projecting BLN neurons in this case. When these red neurons are stimulated, the mouse tends to spend less time in that part of the chamber than the right side. And here's the population data, and sure enough, we found a mild but robust aversion to stimulation of CEA projecting BLN neurons. So now we have these two populations of neurons in the BLA. When we stimulate one of them, we get positive behaviors, and when we stimulate one of them, we get negative behaviors. Two distinct populations, two distinct behaviors. Next, we sought to investigate learning-induced synaptic plasticity onto NAC and CEM projecting BLA neurons. And we used Pavlovian conditioning to study learning-induced plasticity. In this paradigm, a mouse hears a tone, and the mouse learns that either the tone predicts a sucrose reward, in which case it's a positive association, and the mouse responds by consuming the sucrose reward, or the mouse learns that the tone predicts a foot shock, in which case the mouse freezes in fear. Therefore, we hypothesized, based on what I just showed you in terms of populations of neurons in the BLA, that learning-induced plasticity is a function of projection. What do I mean by that? So BLA receives information about the conditioning stimulus onto both NAC and CEM projecting BLA neurons. So, and after the tone acquires meaning, the mouse has to choose the appropriate behavior. So perhaps the way this happens is that after positive association, input synapses onto NAC projectors get stronger relative to CEM projectors, and conversely, input synapses onto CEM projectors get stronger when the tone acquires a negative valence. In order to test this hypothesis, we first injected retrobeads into NAC and CEM. Retrobeads are simply fluorescent tracers that get taken up by axon terminus and label neurons, label all the neurons that send axons there. And then we condition the mice. Mice either learn the association of a tone with foot shock or tone with reward. And one day after they acquire this association, we do in vitro electrophysiology to examine the synaptic strengths of internal capsule inputs onto either the NAC projecting BLN neurons or CEM projecting BLN neurons. And in the in vitro electrophysiology part, we use AMPA and NMDA ratio as a proxy for synaptic strength. AMPA and NMDA are two types of glutamatergic channels. Learning is thought to change the AMPA receptor, the number of AMPA receptors that are trafficked into the synaptic membrane, and therefore, and NMDA receptors are thought to be constant. So therefore, the ratio of AMPA to NMDA is a proxy for synaptic strength. So I have a couple of busy slides ahead of me, so I'm going to orient you to what the slides are going to look like. They're going to look something like this. On the top left corner, I'm going to show you the behavior of the mouse. In this case, the mouse is learning the acquisition of a tone, learns that the tone predicts a sucrose reward. On the right, I'm going to show you the AMP-NMDA ratios, where each circle represents one cell, and each column is represents synaptic strength for that particular group. And I'm going to schematize this, the height of this bar into the thickness of the line here. So let's start. In naive mice, between naive mice and mice which received unpaired tones and foot shocks, there was no significant difference in synaptic strength or AMP-NMDA ratios onto NAC projectors. However, AMP-NMDA ratios decreased when the mouse acquired the association between a tone and a foot shock. Conversely, we use food restriction to motivate the mice to seek rewards. So we used food restricted mice as baseline. And there was no difference in AMP-NMDA ratios between food restricted mice and mice that received both tone and sucrose, but the association was not made. And when the association was made, AMP-NMDA ratios onto NAC projectors increased. Next, let's look at the CEM projectors. The opposite thing happens in CEM projectors. There is a significant increase in AMP-NMDA ratios onto CEM projectors when the mouse associates a tone with a foot shock. And there is a significant decrease in AMP-NMDA ratios when the mouse associates a tone with sucrose reward. So what's happening here? If the thickness of these lines are a proxy for synaptic strength, then after negative association, synaptic strengths are biased towards CEM projectors. And after a positive association, synaptic strengths are biased towards NAC projectors. So I showed you that there are two distinct populations of neurons in the BLA, and they mediate opposing behaviors. And I just showed that inputs to NAC and CEM projecting BLA neurons encode valence. So next we sought to investigate the in vivo ramifications of these changes in learning-induced plasticity. In order to do this, first we hypothesized that there might be an associated change in firing rates in NAC and CEM projecting BLA neurons, which is in line with the changes in synaptic strengths. So if these are cartoon representation of spikes, then perhaps after a positive association, NAC projectors respond more to a tone that predicts a sucrose reward than CEM projectors. And perhaps CEM projectors respond more to a tone that is predictive of a foot shock. In order to investigate this, we expressed channelrhodopsin either in NAC or CEM projecting BLA neurons, and the mice underwent associative learning. In this case, this was a head fix preparation, and the mice associated one tone with sucrose and the other tone with quinine, which the mice hate. So that's the negative association. And then Anna did acute silicon probe recordings in the basolateral amygdala, and after she recorded from many neurons, she used light pulses to determine whether the neurons were projecting to a certain projection target. In essence, whether they're expressing channelrhodopsin. So what did we find? We found that NAC projecting, in response to a sucrose cue, which starts here, NAC projecting BLA neurons fire more as a population compared to CEM projecting BLA neurons. And conversely, CEM projecting BLA neurons respond more to the sucrose cue relative to the NAC projecting BLA neurons. And these are significantly different. So we investigated the role of projection target defined BLA neurons in valence processing. Does it mean that the red cells are essentially these orange cells and the green cells are essentially these blue cells? The answer is no. The simple answer is no, but we found something cooler. So we need to take a closer look at this to actually figure out what we found. So let's say we simplify the responses of a neuron into excitation, inhibition, and no response, no change in firing rate. Then, given that there are two types of conditioning stimuli, there are 9 possibilities of how a neuron can respond to 2 stimuli. These are all the 9 different possibilities. A downward curve indicates inhibition and an upward curve indicates excitation. And the colors represent whether the response is to a rewarding CS or an aversive CS. In order, a neuron is representing valence if we can look at the firing rate of a neuron, and a neuron is representing valence if we can figure out the firing rate of a neuron and look at the firing rate of a neuron, and judge whether the stimulus presented was positive or negative. Therefore, these similar responses we can eliminate as they don't really represent valence. So what I mean by these orange cells is that they show a selective response to an aversive CS, which means it's these two. And what I mean by the blue cells is that they're these. They only respond to a rewarding CS, but not an aversive CS. So what about NAC and CEM projectors? I showed that they are opposing populations, but where do they fall? It turns out that NAC projectors occupy 80% of NAC projectors occupy this bottom right quadrant. And the CEM projectors occupy the top left quadrant. So this is the most simplified I could make this. And this was a huge data set that Anna collected. And there's about 1,500 neurons, and we can sum it up in this diagram where NAC and CEM projecting neurons show opposing responses, but they're not quite the same as these two populations, which we started off with. Okay, so I showed you that projection target defined BLA neurons mediate opposing behaviors, and inputs to NAC and CEM projecting BLA neurons encode valence. And NAC and CEA projector populations have diverging response profiles to positive and negative cues. So I started off by saying that valence is represented in many parts of the brain. I called these two populations as functional populations. Now I'm going to zoom out a little bit, and I'm going to say that what we're doing, I'm going to try to place what we're doing in a bigger context. There are many ways of approaching this problem, and what we have taken is just one of these approaches. So if I try to summarize many possible approaches into one little slide, it's going to look something like this. So we looked at populations of neurons that were defined anatomically, and looked at their relationship to functional populations. Those are not the only ways in which we can define neural populations. Another obvious way of defining neural populations is using genes. So perhaps neurons expressing a gene such as glutamate show different responses from neurons expressing parvalbumin or something like that. And in general, these are what I like to call reverse approaches. And the converse of it is forward approaches. If we actually want to establish the identities of these populations, we could start with them and figure out their genetic makeup and, by comparing gene expression profiles and so on, and look at their projection targets. So that's what I call a reverse approach. And everything I have talked about today is just one aspect, one possible way. So there's a lot of work to be done. And let me try to zoom out in a slightly different way. So these are approaches to studying valence. So what about the BLA? I told you that the BLA mediates aversive behavior and reward seeking. But those are not the only behaviors that BLA mediates. It mediates many others. So is there a common functional role for the BLA across these behaviors? And I'd like to take the next 5 minutes to, so all of what I'm saying now is simply a conjecture. Some aspects of it are tested and it could possibly be true. And this is me trying to place the BLA in a larger context. So perhaps the common functional role is selection. And what do I mean by selection? By selection, I mean that the animal has the task of responding with the correct behavior when the correct cue comes on. For example, when the mouse sees a first shock cue, it needs to take aversive behavior. When the mouse receives a reward predictive cue, it needs to do reward seeking. It's not obvious how this might happen in the BLA. I'll try to break it down into a 3-step process. That's how I think about it. So first, the BLA mediates all of these different behaviors through different projection targets. That doesn't mean that there is one projection target for one behavior. There are many different projection targets. Each of them is specialized in mediating a behavior or a set of behaviors. Now the problem is reduced to something we're familiar with, which we saw in the context of NAC and CEM projectors. So in which case we saw the synaptic strands undergoing opposing modulation to mediate these behaviors. The point I want to make now is that perhaps suppressing the activity related to the irrelevant behavior is maybe almost or even more important than channeling activity into the appropriate behavior. So in order to test this hypothesis, we expressed halorodopsin in CEM projecting BLA neurons and looked at the effect of suppressing this side on reward seeking. And sure enough, we saw that suppressing activity of CEM projectors actually improved the performance of reward seeking in these mice. So the idea of the BLA, so the selection process can be achieved. Perhaps this is a more complete picture, we don't know. Can be achieved by selective synaptic modulation. So maybe a reward predictive cue selectively increases synaptic strength under NAC projectors and decreases everything else. And perhaps an anxiety evoking cue modulates 3 different types of projectors because they're all involved in mediating anxiety. So the question now becomes, how does the brain regulate these 2 sets of synapses? One set of synapses got stronger and the other set of synapses got weaker. And how does the brain achieve this? In order to test this hypothesis, we turned back to our familiar NAC and CEM projectors and went to investigate deeper. We used RNA sequencing to look for gene expression profile differences between NAC and CEM projectors. And essentially what we found was that there were a few genes that were differentially expressed. Some were enriched in CEM projectors and some were enriched in NAC projectors. One of them in particular stood out, which was the neurotensin receptor 1. After confirming that it is indeed expressed more in CEM projecting BLA neurons, we were excited that for this differential expression because neurotensin has the ability to modulate glutamatergic transmission in both ways, increase or decrease, depending on the concentration of neurotensin. So perhaps in the presence of neurotensin, if two different populations are expressing different amount of receptors, we can modulate currents, we increase currents into one and decrease currents onto the other. So Anna did this experiment where she artificially induced LTP into in NAC projecting BLA neurons. And in the presence of neurotensin, LTP was occluded in NAC projecting BLA neurons. and LTP was enhanced in CEM projecting BLA neurons. This is a busy slide, so I'll try to summarize this into a cartoon. So what I'm trying to say is that we have input synapses coming onto NAC and CEM projecting BLA neurons, and there are patterns of activity that are enabling these synapses to change strength. And in the presence of neurotensin, these activities are the activities that are probably common between NAC and CEM projectors are sort of biasing synaptic strength modulation in one direction onto CEM projectors and others onto NAC projectors. Therefore, differential receptor expression is one possible mechanism for opposing synaptic modulation. So, in summary, I would like to put forth the opinion today as that the BLA might act as a selector, and it happens in it possibly happens in 3 stages, or we can think about it in 3 stages. First, BLA mediates various behaviors through its effects on different projection targets. Opposing synaptic modulation can be a key synaptic mechanism for choosing the appropriate behavior and suppressing the irrelevant behaviors. And differential receptor expression between different projector populations could play a key role in achieving this synaptic plasticity. So that's all the science I'm going to talk to you about. And all of the stuff that I've showed you, except the idea of the BLA as a selector, have been published. And as you can see, there are many people who contributed to these works. I did not do all of this alone. And when I look back on and I would first like to start by giving credit to everybody who worked on these projects. So when I look back on the past 5 years, most of the vivid memories that I have are because of the scientific and social environment that you created, okay? So thank you. Thank you for letting me be part of your creation. And when as I said, we had a rough start. But, and I actually wasn't really interested in joining the lab. But then something happened, right? So Kay was really energetic. She was talking to me about these ideas. And then the next day, she comes back to me, scribbles this on her grocery list, and she's like, she used to talk to me about it all day. And that really was the event when I changed my mind and I was in a dilemma. What should I do? So, thank you. Thank you for caring. One day, hopefully, I can become as good a scientist or a mentor as you and create an environment where everybody's supportive and everybody derives pleasure from each other's success. It's time for me. I'm probably better at selfies than Kay. The next person I want to thank is really the person who made all of this possible. None of this would have been possible without Anna. Thank you for being my co-pilot, and thank you for staying with me from the beginning. Because of because of learning from you and working with you, I'm a better collaborator, scientist, and friend. And thank you for sharing your successes with me. And this is the day when the neuron paper came out. I'm also happy. The next person I would like to thank is Gwen. Thank you for being the new team leader of Team Retrobeats. And it means a lot to me that that somebody so enthusiastic is continuing the work that's been done, and it's amazing to have that. And Gwen is also very spontaneous. Well, I'm also very spontaneous. So during this revision periods that Kay described, we were spending 16 or 18 hours each day. And one day, Cheyenne comes up to me and he's like, do you want to go, today is the last day to go sailing. In the moonlight, do you want to go? And me and Gwen were the only two people there. Like, Gwen, do you want to go sailing? Yes. And then I worked with Ada on a bunch of experiments which I didn't have time to show, neural reconstructions, neurotinks and stuff. So thank you for coming to work with me. This was us celebrating Ada's first paper from the lab. And he's in the picture of Ada. Next person I'd like to thank is Romy. Thank you for working with me on thank you for teaching me in Scopics, and thank you for working with me on the rabies virus experiments. And I had a lot of help from undergraduates who are extremely talented. I had the fortune to work with Sarah, Stephanie, and Melody, who were undergraduates who were instrumental in collecting data and also processing all the brains that came out of, 60 perfusions a day. And also thanks to Kim, who was a visiting student. I unfortunately didn't get the chance to present some of the stuff that she worked on. So I would also like to thank our collaborators, Jesse and Susie, who worked on RNA sequencing. Ian Wickersham provided us the rabies virus, and Michael and Reem actually worked with me on the review paper. Next, I would like to take some time to thank my thesis committee. Michael, I've had two long one-on-one conversations with you. One of them made me really want to come to MIT, and the other one helped me decide what I want to do next. So thank you for always inspiring me with the way you think, and I'm really glad you're the chair of my thesis committee. Mark, thank you so much for being a member of my thesis committee. Thank you so much for your constant support. Your letters of recommendation helped me secure funding for the last 2 years. I'm grateful. And I really enjoy our conversations. Now, thank you for being the external reviewer on my committee. I first met you during your course, probably you don't remember, back in fall of 2011. And I really liked your course, and that's when you talked about identifying specific populations of neurons, and I really liked your work, and I'm really glad I got a chance to have you on my thesis committee. And now I'd like the grad school is not just about the science, it's also about a lot of other things. And as one of my best friends, Anna Tavares, said, it's all about the process. And I'd like to take the time to thank the people who made this process an amazing journey. First, the TALA members. Edward, this is you going to marry Lisa. Thank you for being the person who is always on the same boat in graduate school with me. It means a lot. And this is Edward dancing. Thank you for sharing the India-Singapore trip with me, and thank you for baking that cake. Craig? Okay. Well, he'll see the video. Craig, well, I can't address him in person now. I hope to find a lab manager who is at least half as good as Craig, and he's amazing at doing everything. The next person I'd like to thank is Chris. Chris. I started working with you after everything I presented, and I really enjoy working with you, and I think you're awesome, and thank you for doing that. And Tyler, Stephen, Caitlin, Jillian, Tony, Alienor, and Evelyn. Evelyn's back there. Hassan and Gordon, and some of the new people in the lab who I'm hoping to get to know much better in the over the next year. A special thanks to Eyal. I'm glad that I can share my MATLAB geeking out and electronics geeking out with him. And special thanks to Margot for helping me with mouse stuff while I have been busy preparing for this defense. And really, thank you all for all of these amazing memories. This is what I'll remember down the line. So next, I'd like to thank all my. I got the opportunity to mentor several undergraduates and I got a TA. And I also got to spend one semester, 2 hours a week volunteering at a community service center. And you guys are the reason that, part of the reason, a big part of the reason that I decided to stay in academia. And also, next I'd like to thank my friends who I formed relationships with when I came here. My housemates. Past housemates, Zena, well, he's also my housemate now, Valerie and Melissa, and my current housemates, Galen, Max and Julian, and my friends, Emily, Cheyenne, Swati, and Anu, and my partner Lena. And finally, I'd like to thank my family, especially my mother. I'm only because of her hard work, dedication, and sacrifice, and none of this would have been possible without her. And thank you all for listening. I'm happy to take questions if you have any.
So the two types of projection neurons in PLA, are they interconnected, and does that circuitry allow?
Excellent question. Yes. So I think there's when we are thinking of the BL as a selector, I think there is a unique position that BL occupies, because all of these projection target neurons are topographically intermingled, and in general, if you stimulate neurons within the BLA, you're going to see excitatory and inhibitory responses from other neurons. And it would be really interesting to see if they could implement something like a winner-take-all mechanism, where and total activity in one population is what determines which population wins. So that's an excellent question.
All right, if there are no further questions, then we'll just thank Praneeth for his presentation and proceed to the closed part of the defense.